students. So, we are continuing our learning in immunity, how you can boost your immune system, and how lots of immunogenic products are nowadays in market. And this is the last lecture in our understanding of immunity and immunology. So, today I will end up my lecture, my entire series of lecture with the blood products and plasma substitutes. So, I am going to tell you about the various kinds of blood products collection, how you can collect say various types of bloods. I will also tell you the dried human plasma. Then I will also tell you how you can process and storage the dried human plasma and other kinds of blood product. Let me tell you before discussing, let me tell you some basic understanding. So, blood it is a connective tissue consisting of your plasma and cellular component like your red blood cell, RBC and platelets. If I show you in a pictorial form, I think in my previous lecture I also have discussed with this slide. So, you can see this is our blood vessel, okay. It might be your artery or it might be vein. So, in that blood vessel you have blood. So, in that blood you have the RBC, red blood cell, you have WBC, these things there are various types of WBC which are actually helping in our immunity, neutrophil, eosinophil, basophil. We have seen in our previous lectures that how they are actually helping to boost up our immunity. Then we have various kinds of platelets which are also helpful, and another most interesting thing is called actually plasma, this yellow part, okay. This yellow part actually plasma. Based on that plasma we can develop another technology, another therapy called plasma therapy. Now, let me tell you about the differences. I think in this part also I have told you that what are the differences between serum and plasma. So, it is very simple. If you just eliminate the clotting factor from the entire serum, for example, this in that yellow part, if you in that yellow part, this is called in total it is called plasma. So, in plasma you have serum, you also have some clotting factor. If you just remove the clotting factor, you will be getting the serum. So, what are the activity of various kinds of the blood cells? So, the normal blood cells which are RBC, WBC or platelets, they can do lots of work like these things. They can supply oxygen, the RBC, the granulocytes are actually help in that is ingestion or destroy the pathogens. Monocytes are actually defend against infection. Lymphocytes are actually that is helpful in the immune response like B cells and T cells. Plate cells are actually helpful in bleeding, platelets. And plasma, the most important, they are doing lots of work like lots of proteins are present in plasma like albumin, immunoglobulin, fibrinogen. Albumin it is actually maintain the osmotic pressure and also transports various kinds of substances. Immunoglobulins they regulate immunity and the complement system which help clear to the pathogen. If a virus is actually entered, the immunoglobulin, that is antibody whatever we have discussed in my previous lecture, they will come to the picture, they will come to the that is a area of infection and it will take some action. Fibrinogen, they are blood clotting, that is during blood coagulation they will do some their work. And in other plasma there is also some other work like they also helps stop bleeding, maintain the blood volume. So, this is the entire picture that how blood is important and how various that is components of our blood they are doing lots of tasks to maintain the normal activity in our body. Now, let me tell you that in case of donor and recipients we already have discussed that we cannot that is receive blood from anybody. Why? Because of the various kinds of blood group. How it is actually grouped? It was grouped based on their antibody present in the plasma and antigen present in our RBC. This is our RBC and you can see these antigens. You can see these antigens. Okay. You can see there is no any antigen is present. So, based on these antigens our all the that is blood groups are actually developed. So, if A antigen is present, then A, B antigen is present, then B. If both are present, you can see both are present, then B. If none is present, here is no any antigen, so it will be group O. Due to that differences, its opposite antibody in the plasma will be there. You can see opposite, A means anti B, B means anti A, opposite. So, based on that, our this chart was actually developed, that who can who are the donor, and if they donate, who can accept this blood. Okay. So, in nutshell, everybody cannot donate blood to everybody. We have to follow the chart. Now, after learning the basic concept in our blood's component, and who can donate blood to who, and how that is our various blood cells are actually taken lots of work. Now, let me tell you, when if you organize one blood donation camp, or if you are happy to donate your blood, then how the activity will happen and during that is a blood product collection, what should you do, we should do, and how you may proceed. So, first let me tell you that abstracted donor, the one who is not suffering from any disease transmitted by transfusion. This includes syphilis, malaria, or serum jaundice, or any kinds of anemic. If you are completely free, if you are completely that is a fit, if you do not have any kinds of these diseases, syphilis, malaria, serum jaundice, and you are well, then you should be you can act as a donor, abstracted donor. Collection the blood is collected from that is a median cavital vein in front of elbow into a sterile containing anticoagulant solution, because in your blood you already have that is a blood coagulating factor. So, what will happen if you just collect all the bloods in a container? Okay, what will happen? Because of the clotting factor, entire blood will be get clotted. That is why in that container there will be some anticoagulant solution. So, you should collect this blood and you may store in that that is a storage part. Now, how much blood you may take or how much blood you may collect? Not more than 420 milliliter taken at a once one attendance immediately. Afterward, the container is actually sealed and cooled at 4 to 6 degree centigrade. Okay, because at this is the optimum temperature. At that temperature, it will be not very easy to drugs to bloods to get degrade. You have to do some test. Okay, you have to do some test after storing. One is the serological testing to confirm the absence of various kinds of diseases, and also you have to do some that is a blood grouping, that is AKtU grouping, that is a test to see or to understand or to determine the group of the blood, whether the person having that is a group A, group B, C, whatever, sorry, not C, O and AB. So, you have to do two kinds of test for the if there is any disease person has given the blood, or you have to check the group of the person has given the blood. Then you have to store at the 4 to 6 degree centigrade, already I have mentioned. Then where you can use? You can use in case of that is severe loss of blood from the body during hemorrhage, shock, or uncontrolled diarrhea or vomiting. Any of the cases, other diseases are also that is possible where you can use this whole blood. Now, apart from understanding this part, let me tell you about the dried human plasma or also whole human blood. So, the whole human blood, whatever I have mentioned, I have shown you here, there are some disadvantages. It has poor keeping properties, necessities for that is within that is 3 weeks. It requires refrigerated storage. You have seen here, I have mentioned that 4 to 6 degree centigrade, and it must be compatible with the blood of the recipients. That is the major disadvantage of the whole blood, whatever I was actually trying to mention. So, you might be you might have heard that lots of organizations, voluntary organizations, they are organizing with a very honest that is thought some blood donation camp. But the problem is that if they do not able to store it proper manner with maintaining the temperature and other kinds of storage condition, what will happen? The blood will be not kept in a proper manner, it will be become some toxic or other things. That is why storage is very very important. Another thing, you have to store in this, okay, 4 to 6 degrees centigrade. The problem in India is that, or other kinds of equatorial or Mediterranean countries, high temperature, and it is not very easy to maintain the low temperature because of the lack of electricity in the rural areas and lots of problem. So, that is why we have seen it is a big challenges, it is a big challenge in front of us in regarding to keep and storing the whole human blood. So, that is why we have to think differently, we have to identify something else. So, what is the new technique or how we can solve this technique? The answer is dried human plasma. Dried human plasma, this is the picture. It has a lots of advantages. Instead of whole human blood, you can give this. So, properly it can be stored and kept for at least 5 years. If protected from light, it can be stored for at least 5 years and temperature provided value of the 20 degree centigrade, and it can be given to the patients of any that is a blood group. So, obviously, it has a lots of advantages. Okay. So, here you can see 20 degree, below 20 degree. So, at that temperature you can keep this, but in case of our actual blood, we have to that is store it at 4 to 6 degree centigrade. So, obviously, in country like India or Mediterranean country or like equatorial country, it is not easy to get such kind of temperature, that is low temperature. So, we have to take the help of the electricity and air conditioning and other kinds of facilities to maintain at 4 to 6 degree centigrade, but 20 degree centigrade, it is not a very difficult task, we can get it, we can manage. So, that is why dried human plasma has lots of advantages. So, if I tell you the major problems to overcome for the preparation, that is a transmission of viral jaundice by pooling, neutralization of the plasma agglutinin by cross that is naturalization by soluble agglutinogens, and most satisfactory ratio is the 9 of A, that is 9 of that is O 2 of B or AB. So, this is the ratio how you can give. So, how you can process or you can store the dried human plasma. So, dried plasma is prepared from the time expired circulated blood, which has been centrifuged to deposit the cells. More than 40% of the supernatant fluid is actually siphoned off through the sterile tube, and after that then you pooling is done, and then correct ratio of the blood grouping chosen to neutralize the powerful agglutinogen, then separate that is testing for the sterility are to be done. Then 400 ml of that is quantity is actually dispensed in MRC bottle are then subject to the freeze drying, follow this word freeze drying. Okay. So, after that yeah, now I have to tell you that in case of freeze drying, that there is something called primary freezing, and I also I think mentioned the secondary that is drying. So, let me tell you the primary drying. So, the bottles of frozen material are actually mounted in the horizontal in drying chamber, and high vacuum is horizontally in the drying chamber and will be applied. Okay, high vacuum, and your after that the ice sublimes on the condensing coil kept at minus 50 degree centigrade, and a that is small hot heater provides the latent heat which will be required for the evaporation of the certain substances. So, this stage takes about 2 days, and after which the residual moisture content is about 2%. So, you can see only 2% residual actually we are actually getting. So, lots of wastage actually happen. In secondary drying, it is done another chamber by vacuum desiccator over phosphorus pentoxide. It takes about a day and the product is left with the 0.5% moisture, and then storage it should be kept in the value 20 degree centigrade. We have seen in earlier slides that 20 degree centigrade is the appropriate temperature when you can store it, and protected, it should be protected from the moisture. There are lots of reasons why you should keep from moisture, including sunlights and the other things. You see, if moisture may present, what will happen? Lots of microbes and other things can grow in the moisture. If there is sunlight, so because of the presence of light, there are some photogenic reaction might be possible, and if the high temperature is actually coming, then there are chances the breakage of bond in that particular that is a substance. So, that is why you have to keep them away from moisture, sunlight and other things, and it will be remain usable for 5 years. But the problem is here that we will be getting actually very less amount or small amount of the dried human plasma. So, where you can use it? It is satisfactory alternative of the whole blood in condition where there is no loss of RBC, that is red cells. For example, like burns or scalp, where there is a excessive fluid or protein actually loss. In case of the emergency, whole that is human blood is not available. Somebody is having somebody is not getting the blood. So, nowadays what we do? We just post one Facebook post or Twitter, or we just that is update some status in WhatsApp that somebody is needing some blood, some group of blood in one hospital. If you are having this blood group's blood, then please you may go there and donate the blood. But in every time when we should actually we are not lucky. So, in such cases where there is no blood is available, in that case whole that is this technology that our dried human plasma can be an alternative. Okay. When this whole human blood is unavailable, then our dried human plasma will be a great alternative. So, if I tell you other products in this area. So, yeah, in case of preparation, I already mentioned that how you can prepare it. I have actually tried to summarize everything. Wait, some problem is happening. Yeah, I have actually summarized everything in a very systematic manner. So, it is very simple. You may allow it to clot, and after that supernatant should be collected, and you may keep in this temperature, and you can use when burn and other things are available. So, here is a very good picture. You can see in this container our dried human plasma actually they have saved. This is a human plasma protein fraction. So, for human plasma protein fraction, how you can produce? So, you have to for the preparation, the fraction of pooled citrate plasma not more than 0.4% and organic solvent actually used having proper volatility or bacteriostatic that is activity. Bacteriostatic, you may understand bacteriostatic, which will be against the bacteria. A stabilizer such as sodium platelet host fraction is to allow the preparation to be heated for hours at the low temperature without any degradation of the protein. Where you can use in case of fibrinogen deficiency, and in case of also in conjunction with the that is thrombin to assist the adhesion of the skin graft. Another plasma substitute, the limited supply of the plasma at the cost of the producing of dried form. The risk of transmitting serum heavily simulated atoms to find substitute of non-human origin that could be used to restore blood volume temporarily, while the recipients replace with the lost protein, for example, gum saline and polyvinyl that is pyrrolidine, these things you can use. Okay. And if I tell you more, what will be the ideal properties of the plasma substitute. So, same colloidal osmotic pressure it should possess, and a fairly low rate erection if it is possible, and freedom of the from toxicity. It should not be toxic, it should not create any harmful effect to our body, okay. And it should be free from antigenicity and pyrogenicity. See, antigenicity again our concepts are actually coming here. It should not be antigenic. If it is antigenic, then your entire that is immune system will become active, then they will come to the picture, they will take the action. So, it should not be that is antigenic, it should not possess any kinds of antigenicity, and your immune system may accept this thing, your immune system may accept this thing as a part of our human cells. Okay. And if I tell more about the ideal features, so high stability in liquid form at the normal sterilizing temperature during the transport and storage, and ease of preparation, ready availability at low cost, very important, readily available and low cost. See, actually if I tell you honestly, still the date we are so much relying on our conventional procedure because of the cost effectivity and that is a ready availability, and molecular weight such that molecule do not easily diffuse through the capillary walls. So, you know Lipinski, again I am bringing the medicinal chemistry concepts. Lipinski told, if a substance may work as a drug, its molecular weight should be less than 500 Dalton. Okay. But is it possible to make every molecule less than 500 Dalton? It is not easy. So, that is why we have to think in such a manner, because ultimately those components it will be that is spread throughout our body, and the capillaries and other things, their size is very small. So, it should be compatible with that particular area, it should compatible to go with those areas. Okay. Now, before ending my lecture, let me also tell you that in this case, like plasma substitute or whatever we have seen, that means dried human plasma and other things. So, you have to that is we need to adopt the new technologies, okay, because in future you see we have a lots of challenges. In future we will be observing that lots of new diseases, because in last year in 2020 there was 2 news reports. One news report was actually from Russia, and another was not only from India, entire global media actually covered. In Russia, the Arctic Ocean and those Iceland's area what is actually present, due to global warming, ice are actually getting melted. Okay. In our that is Himalaya, all those glaciers are also getting melted. The problem is that recently in 2020, last year, lots of animals like cow and goat and other animals, they actually died because of some unknown reason. When scientists actually went there to check the reason why that is unreported or unknown cause they are actually dying, they have found that they actually died by some infection related to virus. Okay. These are the new viruses which was never reported. Now, when they have done more study, they have identified that during the ice age, I hope all of you are aware about the ice age, almost 10 million years ago happened in our earth. So, during the ice age, whatever the viruses was there in our environment or in our world, they are actually got trapped within those ice, which are actually kept in the form of glacier and other forms. Okay. So, due to global warming, now those ice are actually getting melted and new viruses are actually coming, and these viruses are actually infecting to the domestic animals or your cow and other things in the Tibet and other Russian area. So, that is a very major challenge in front of our scientists. So, people are predicting COVID-19 will be not the last that is a pandemic. That is why CDC in US, in our India also ICMR and other organizations, they are doing tremendous work how to save human from the new viruses. What is the solution? Solution is very simple, whatever I have told in my last 10 lectures. Solution is immunity. We have to boost up our immunity, we have to train our immune system with new viruses, we have to make new vaccines, and we have to work with newer technologies like monoclonal antibodies or like hybridoma technology, so that we can make monoclonal antibodies, or we have to work with newer forms, so that we can fight. So, that is why our this understanding of subject is very important. So, thank you all of you for listening my lectures. since last many days. So, God bless you all. I hope you will do something great in the field of immunity to save our immunity for in the future challenges with viruses. Thank you. God bless.